growth promoting fungi, or simply known as PGPF. These fungi are not enemies of the plant. In fact, they are non-pathogenic, forming a symbiotic alliance with a plant root. Through this intimate association, they are well aware of support and nourishment. Their role in agriculture. PGPF enhance the nutrient uptake by transforming inaccessible compound, and they also produce phytohormone, which is a natural growth regulator and as a natural decomposer. PGPF, such as Trichoderma, Penicillium, and Aspergillus, play a vital role in sustainable farming. They fight off plant pathogens through antibiosis, competition, and mycoparasitism. With PGPF, we reduce our reliance on chemical fertilizer and pesticide, improve soil health, and support long-term food security.
Plant growth promoting fungi play a vital role in enhancing nutrient availability to plants, particularly by solubilizing essential nutrients such as phosphorus, potassium, and zinc. Nutrient availability in the rhizosphere is controlled by combined effects of soil properties, plant characteristics, and root microorganism interactions. Plant growth promoting fungi can enhance nutrient uptake and availability. For the phosphorus solubilization, plant growth promoting fungi produces organic acids such as malic acid, gluconic acids, and enzymes phosphatase convert insoluble phosphatase into plant available soluble forms. Key fungi involved are Penicillium species. For potassium solubilization, fungi solubilize insoluble potassium bearing minerals through acid production and chelation, improving the uptake of potassium by plants. Key fungi is Aspergillus species. For zinc solubilization, plant growth promoting fungi enhances the solubilization and plant uptake of zinc through mineralization and chelation processes. Example of fungi is Trichoderma species.
Stimulate different forms of defense mechanism to resist disease by invasion of pathogen. Plant growth promoting fungi like Trichoderma species enhance plant immunity through induced resistance or activating defense pathway without direct pathogen contact. Induced resistance is a distinguished process of promoting the immunity of plant. There are two types of induced resistance: induced systemic resistance (ISR) and systemic acquired resistance (SAR). ISR, or induced systemic resistance, is a process that involves the inhibition of disease invasion by pathogen in plant. How ISR is activated? PGPF such as Trichoderma species colonize the plant root without causing a disease. This root interaction acts like a beneficial stress that primes the plant immune system that could activate the JA, jasmonic acid, and ethylene cytidine pathway that leading to ISR. Systemic acquired resistance (SAR) is a form of induced resistance that expresses a discrete signal transition pathway. SAR is activated after localized pathogen infection or exposure to synthetic compounds. Some PGPF can also stimulate salicylic acid production, especially when they induce a mild plant stress or localized infection like responses. This could trigger SAR and a defense response usually activated by a pathogen attack. It may lead to the expression of pathogens-related PR proteins such as PR1, PR2, and PR5. There are several differences between induced systemic resistance and systemic acquired resistance.
Beneath the soil surface, plant growth promoting fungi, or PGPF, play a critical role in supporting plant development. These fungi live in the rhizosphere or even inside plant tissues, where they influence growth through several biological mechanisms. One major contribution of PGPF is their ability to produce plant hormones, particularly auxins and gibberellins. Auxins, like indole-3 acetic acid (IAA), are commonly produced by fungi such as Trichoderma and Aspergillus. These hormones stimulate lateral root formation and root hair development, increasing a plant's ability to absorb water and nutrients, especially under environmental stress. Gibberellins, made by fungi like Cladosporium and Gibberella fujigoroi, promote seed germination, stem elongation, and overall biomass growth by triggering changes in the plant's growth regulating proteins. Together, auxins and gibberellins coordinate plant architecture, making the root and shoot systems more efficient. Their natural production by PGPF is especially valuable in sustainable agriculture, reducing the need for chemical fertilizers and helping crops perform better in challenging environments.
Plant growth promoting fungi also play a role in soil structure and microbiome modulation. PGPF helps soil stay together by forming tiny networks that hold soil particles, which increases the amount of space available for water and air. They also affect the soil microbiome by promoting good microbes and decreasing toxic ones. Plant roots are better able to develop, absorb nutrients, and withstand stress as a result. In soil structure improvement, PGPF produce extensive fungal hyphae that bind soil particles and produce stable soil aggregates. For example, glomadine produced by mycorrhizal fungi help to keep the soil stable and full of air and water. Not only that, in soil microbiome, PGPF affects the microbial community structure surrounding roots, rhizosphere, by opposing pathogens and bring nutrients to the roots. For example, Trichoderma species release enzymes that promote beneficial microorganisms and inhibit fungal pathogens. By improving the soil, it's help root growth and protecting the roots from disease.
Hi, I'm Afni. So I will explain these two topics. Real world application. Sustainable agriculture. Reduce non-chemical fertilizer, potentially enhancing the plant growth. Next is biological disease control. Some act as biocontrol agent that harmful the pathogen through enzyme secretion. Lastly, soil rehabilitation and health. It improves soil structure and microbiodiversity and improve organic matter content in agricultural ecosystem. Sustainability content. Reduce environmental pollution. By replacing synthetic agrochemical, PGPF prevent water and soil pollution. Next is promote biodiversity in soil ecosystem. Encourage microbiodiversity and healthier soil microbiome, which are vital for long-term soil fertility and ecosystem stability. That's all from me.